The society has for a number of years, every so often had its president or some of its senior members meet with NIH, talk about where the field is going and discuss how we might move things ahead. And at the beginning of last year, 2010, when Ken Cornetta was president, I was president-elect, there was a particular opportunity because in the 2 or 3 preceding years there'd been some very clear positive events that happened in gene therapy trials, particularly in the immune diseases and also in the retinal degeneration disease. There'd been some very nice clinical results, and these are the first sort of really strongly positive clinical benefits that came out of that, and it became clear that gene therapy could really improve quality of life in patients. And actually those events got some enormous public coverage, and so we thought that was a wonderful opportunity to meet with each of the directors, or as many of the directors at NIH as we could, to discuss the fact that there was beginning to be this positive accumulation of results, and how could we take advantage of this best to really move this field forward now. Because once you begin to see what a technology can do in the clinic, it becomes in a sense easy to plan how to move it forward. It becomes clearer kind of what resources we really need or what might be holding it up. And also this has come on the top of 20 years of clinical trials for gene therapy. The first one started actually 21 years ago now, and those trials actually began at NIH, so there's been this big investment. Nevertheless, so Ken Cornetta and I spent a couple of days. We did something like 14 one-on-one meetings with nearly all of the individual institute directors, a number of center directors, and discussed with them where the field was, and discussed with them some of the concerns we had, and actually there were concerns collected from our members. Difficulties of getting coherent funding, stepping through from seeing a scientific proof of concept in animal models and how to progress to the clinic and be able to generate vectors and do the preclinical things and interact with the FDA. It's hard to assemble that funding. It was hard. Some of the institutes were very familiar with what gene therapy was doing because they were particularly involved in funding. Other institutes were much less familiar because they had not actually been directly funding gene therapy. And so nearly all of these directors asked what they could do and asked what we would do, and most of them felt, and some quite explicitly said, this would be great if the society would come, mount a workshop, mount a meeting at NIH, and just to describe where we are now, what we know about this field, this technology, the really nice results that are coming, and talk about how we can expand that, talk about what are challenges, what is perhaps holding back the field, and also a couple of the directors quite explicitly asked, could we challenge the NIH into things that we would like to see them do. So I think that it became very clear that both to educate some of the program staff who have to make funding decisions, and to really get everybody up to speed, that the idea was to hold exactly this session. So it's educational in the sense that it's educational for people in the field and people in the funding agencies as to what we've really achieved, and they're pretty major achievements, right, but it's barely the beginning. Right. And so now the goal is to how can we best direct what funds we have and what efforts we can mount to really take bigger advantage.